Now the participants are joining, so we will start at 3:05. Okay. We are sharing the link.
Sir, Rama Shankar Singh.
Yeah, okay, sir. Welcome, welcome.
No issue, sir. कोई दिक्कत नहीं है. 4-5 मिनट में कोई फर्क.
Because participants are joined, we just share the link of the meeting. Okay.
Okay, sir. No issue, sir. No issue. Okay.
Okay. Ma'am?
Yes, sir.
So, shall we start?
Yes,
so,
you are listening me?
Yeah, ma'am, I listening to you.
Okay. You start. You can start.
Yeah. So, dear participants, good afternoon to all. Today our notable speaker is Professor ALKA from Indian Medical Institute of Banaras Science, Banaras University. As we know that the medicinal chemistry is an important area. Medicinal chemistry is an important area for in the medical field. So as we know that there is a lots of chemistry are using in the medical field, because we know that without chemistry there is no medical science. Because we know that the basic science is important in every field, even though in aerospace, even though in medical field, even though in engineering field, basic science is very important. So now today our session is medicinal chemistry, because we know that the medicinal chemistry has an important area in the field of medical field, because without chemistry there is no medicine. Actually, do you know that we have studied the lots of molecule in the chemistry? So how such things happen? This is due to the chemistry, because the chemistry is used to study the small molecules. Without medicinal chemistry, we can know and that this compound is important for the purpose of the allied subject, diamonds, or the any disease. So my voice is audible. At then
Yes, sir, you are audible, sir.
there is no problem. Okay,
No
okay,
issue, sir. Very clear. Voice is clear.
okay. So the chemistry is very important subject in medicine. Major contribution to the health care has been chemistry. The development of new drugs involves chemical analysis and synthesis of new compounds. Because the new compounds we don't know about that these compounds may be fruitful to some disease or that particular disease or this may be fruitful to our health issue. So the chemistry studies the lots of compound and say about that these compounds are important. So many recent television program advertise the large number of new drugs of corona. You know that Baba Ramdev also the coronary and other drugs, Ayush Ministry, these are. So how this happened? Because the on this basis the chemists are behind on these drugs. So the medical chemistry is very important. The development of new drug is long and complicated. As we know that it is almost 1 year has gone. There is no medicine for the COVID-19 cure, and it means the medicine has a very long and complicated time. The chemistry of disease must be studied as well as how the drug affect the human body because the most of the drug have this side effect, and if they have the nominal side effect there is no problem. So a drug may work well in animals but not in human beings. As we know that some drugs may work in animals but not in human beings. Out of 100 drugs that look like that help treat disease, only a small handful actually turn out to be the both safe and effective. As mean that there are a lots of vaccine pass crosses over the third trial phase, but nobody has said that this will be effective and this is having no side effect. So that is an important area, and such area is coming among the medicinal chemistry. The chemistry contribute the preparation and use of materials for surgery, and the structure used in many surgery today do not have to remove because they simply dissolve in the body after a period of time. The replacement of blood vessels from heart and other types of surgery are often made of chemicals that do not react with tissue, so they will not be rejected by the body. So, how the chemistry is useful in medical field? So, there is a large number of criteria. The medical chemistry is useful in the medical field. First, chemistry finds many applications in healthcare field. Development of medicine involves many complicated chemistry process. Chemistry is used to create material used in surgery. Much laboratory testing is based on chemistry section. So, participants, we have know that the medicinal chemistry is a very important topic today. So, we have invited the Professor ALKA Agarwal, Indian Institute of Medical Science of Banaras Hindu University. Do you know that? This institute is very reputed and it is equivalent to AIMS. So I brief about the Professor ALKA Agarwal. She is a professor in Indian Institute IMS VHU and she is also presently head of this department. And she is MSc Chemistry and PhD from the Awadh University, Faizabad, UP, and she also got a international award, academic research fellowship award, INSA Exchange Visiting Science Award at Ruhr University, Bochum, Germany, INSA Exchange Visiting Scientist Award at Indian Institute of Science, Bangalore in 2014. He has also have a various administrative experience in Banaras University, like she has a chairman of Department Research Committee, Department of Medical Chemistry, BHU Banarsi, Chairman of Board of Research Studies, BHU Banarsi, She is also member secretary of Department Research Committee 2017 to 2009. She is also member secretary of Department Purchase Committee 2010. She also examiner in practical and bio examination of different reputed university in India. She is also chairman of Department Research Committee 2018 to till date, chairman of Department Purchase Committee 2018 to till date. She is also examiner in South Campus RGSC BHU Banansi. It is also a part of BHU. And she also administrative expert outside Banaras University. She is expert in UPSC Assistant Professor Framing Guideline Committee. She is also subject expert in Selection Committee of Assistant Professor, Associate Professor in Mondin Chipchi Master Vishwavidyalaya Lucknow UP. He is also having the various assignment outside the BHU, like steering committee member, secretary of Ayurveda Pharmacia Committee 2007. She is also observer in UGC NET, G, CTET, NEET examination organized by CBSC New Delhi since 2013 to till continue. She also is a members of various reputed national and international professional bodies such as she is also life member of Indian Science Congress Kolkata. She is also life member of International Academy of Physical Science Allahabad UP. She is also member secretary and Indian Society of Promotion of Education and Science Delhi. She also contributing in the field of research, and she has supervised the almost 10 students, and almost 4 students are undergoing to submit their PhD thesis. She published research articles in International Journal almost 40, as well as in National Journal almost 20, and 2 book chapters. And she also filed the 9 patent. That is a huge thing that in the Indian national patent. And she also has the H index 12 and having the various experience in abroad as well as in India, like the Central CRI Lucknow and Central Council of Research in Ayurveda and Siddha Government of India, New Delhi. So this is the only brief about the ma'am, but she has lots of award and lots of research in this area. So welcome, ma'am. Welcome you again, ma'am. ALKA, ma'am.
Oh, yes.
So I am handed over to you. So participants, listen carefully and mute their mic on the conference. And okay, ma'am.
Okay. Thank you, Dr. Lakshman Singh, giving such a nice introduction about my candidature. Today I will talk on the synthesis of the diversified antimicrobial agent using click chemistry. This work is going on in my laboratory. My main research interest in my laboratory are going on is catalysis, synthesis of peptides, design and synthesis of antimicrobial agents, modification in drugs and nanoparticles. All these 5 research is going on 5 topics, but today I will discuss only on the design and synthesis of antimicrobial agents and nanoparticles. These are some drugs which are as in the market, which is used as an antibacterial or antifungal. These are the anti-tumor drug which contain this benzothiazole moiety. This is used as antibacterial, anti-tumor drug. This is the oxygen receptor antagonist which have also the benzothiazole moiety. And this is a drug which is contain also the benzothiazole moiety. It have the gram-positive selective antibacterial drug which is used in the market. And this is the fluconazole. It contains the 1,2,3,4 triazole nucleus. This is albuterol also contains the triazole nucleus. And this is the voriconazole also contains the triazole nucleus. I am telling about these drugs because these molecules are very active, having the basic nucleus of this benzothiazole and the triazole, which is responsible for the activity of these compounds. When we think about the development of a drug, we have to some points always in our mind that the drug development should be the synthesis should be easy, it is feasible in the lab, and the intermediate and the final product should be cheap. If it is costly, it is very difficult to manage in the lab. And the synthesis process should be eco-friendly. We are not using the hazardous solvents or chemicals. And the final product should be formed, it should be negligible in toxicity. These are the some work which is going on in my laboratory, and we have published the several papers on international journals. This is the first paper. This is where we have synthesized the new antimicrobials and characterized by the NMR and crystallography. This paper we have published in European Journal of Medicinal Chemistry in 2016. And this is a pure catalysis paper in which we have to develop the tetrazole by acetonitrile, a substitute acetonitrile in one pot synthesis and characterized by the IR, NMR and the XRD. This paper we have published in 2015 in the CR Chemie. And this is a theoretical paper. One of my students is working on the theoretical papers that our compounds are showing the good activity, how it is possible. He is doing the Hirschfeld surfaces and DFT computation of novel sum compounds, active compounds. And this paper we have published in the Journal of Molecular Structure in 2017. Now come to that we have synthesized a lot of compounds, and our compounds are showing very good antibacterial activity. And this activity are sometimes this MIC are less than the standard drug ciprofloxacin. Sometimes it is just half, or sometimes it is 1/4. MIC is 1/4. So we thought that how our compounds are working with the human serum, human body. So we have tested our active compounds with the HSA, human serum albumin, by the different techniques, and then we found that our compounds showing the better activity with the in comparison to the standard drug. And this paper introduction of the antibacterial drug, which is synthesized in our lab with HSA, and published this paper in RSC Advances 2019. Now we have recently started the work on the nanoparticle because there is a boom of nanoparticles. So we have synthesized some novel sulfonic acid supported magnetic nanocatalysts and characterized by the TEM, SEM and PXRD and then the IR, and we used this nanoparticle after characterization. We have applied this nanoparticle that how it can be worked. It is used in the formulation of the primary amine formulation into the primary amines. And this paper is recently published in 2020, 1 month before, in RSC Advances. This is a summary of work which is going on recently in my laboratory. These are the some antibacterial compounds which have been synthesized in our lab because we are every time searching the new molecules which is active in comparison to the ciprofloxacin. These are the some peptides we are using. I have already told you that this is a benzothiazole nuclease which is in the market, right? So we thought we synthesized some derivative of this using this benzothiazole moiety, and we synthesized the peptides. This is a dipeptide, lysine and valine, and this is the lysine and the glycine, and tested this compound against bacterial strains. We found that our compounds are showing the MIC at 5 microgram per ml. Both the compounds are showing the same MIC. And this is another compound which have the ethophenylene diamine derivative, and we are using the triazole nucleus, and we found that our compound are showing the antibacterial activity which is similar to the market drug ciprofloxacin. This is 6.25 microgram per ml. Now this is some chalcone derivatives. Chalcones are always the free radical scavenger, so we thought we synthesized some chalcone derivative, and we tested against the gram positive and gram negative bacterial strain, and these compounds are showing the very good antibacterial activity which is similar to the standard drug ciprofloxacin. This is 6.25 microgram per ml. And this is the another compound. This is the benzothiazole nucleus containing compound having the 2-triazole nucleus, bis triazole containing compound. This compound is showing is a potent antibacterial agent, and this is showing the antibacterial activity 3.12 microgram per ml, which is just 50% of the standard ciprofloxacin. Ciprofloxacin shows up antibacterial activity at 6.25 microgram per ml. And this is the paper. All the compounds are published in the different papers. This is European Journal of Medicinal Chemistry 2013, and Medicinal Chemistry in 2014, and Medicinal Chemistry, European Journal of Medicinal Chemistry in 2016. These are some other compounds which we have published, prepared in our synthesized in our laboratory. This is again another chalcone containing the bis triazole nucleus, which is also showing the very good antibacterial. This is similar to that ciprofloxacin. This is the chalcone. From chalcone we have synthesized the flavone, and this flavone is also showing the very good antibacterial, which is similar to that of ciprofloxacin. This compound and this compound are showing the very good antibacterial similar to that ciprofloxacin. This is the tetrazole and triazole containing nucleus compound. They are a very potent antibacterial agent. They are showing the antibacterial activity at 1.56 microgram per ml, which is the 1/4 of the activity of the ciprofloxacin. And this is the another compound, tetrazole and triazole, which is also showing the just half MIC of the standard ciprofloxacin. This paper is also published in European Journal of Medicine 2016. These are this is a drug modification of the existing drug ciprofloxacin. This is a ciprofloxacin nucleus. We have modified the ciprofloxacin by adding these two triazole nucleus and tested for antibacterial activity. This compound are showing the four times more potent than the ciprofloxacin, is 1.56 microgram per ml. And again, these compounds are a similar compound, but this nucleus is changed. This is a chloro group. Here is a bromo, and this is the position of the chloro group is changed. This is the 3 position, and this is chlorofluoro. This is the orthomethoxy group, and these compounds are showing the very good antibacterial activity against the bacterial stains in compared to the ciprofloxacin. This compound are the most potent. It is showing the 0.78 microgram per ml. This paper is also published in European Journal of Chemistry in 2016. These are another compound which have again we advertise the ciprofloxacin nucleus. One position is blocked by the ethoxy, methoxy, propoloxy, ethoxy, and methoxy group. And here is we have added the triazole nucleus. And these compounds are showing again the very good antibacterial activity in comparison to the ciprofloxacin. This paper is under revision in European Journal of Medicinal Chemistry. This is a click chemistry. We are developing our triazole nucleus by the click chemistry. What is a click chemistry? This is 3+2 cycloaddition. This is azide and alkyne cycloaddition reaction. This is click chemistry is developed, introduced by the K.P. Sharpless in 2001 to describe reactions that are high yielding, wide in scope, create only byproduct that can be removed without chromatography, are stir specific, simple to perform, and can be conducted in easily removable solvent. It means that only one product is formed and eco-friendly, in which we have the eco-friendly solvent we have used. But before that, there is another process by preparing this triazole nucleus. There are two residual isomers formed, which is very difficult to separate. So actual click chemistry is not performed in this reaction. So we have used this process to form the triazole nucleus in our compounds. This is the copper catalyzed. We have used a copper 1 catalyst, copper 1 salts, and in azides, and this alkyne, and we got this 1,2,3 triazole 1,4 substituted disubstituted triazoles in our compound. This is a mechanism how this catalyst act, this copper sulfate salt act and form the triazole nucleus. This is by using this click chemistry and using this benzotriazole nucleus, we have synthesized our compound, which I have already told you that our compounds are showing the very good antibacterial activity. This is benzotriazole derivatives, where we have started with the 2-amino benzotriazole nucleus, which is propagated by the propyl bromide in presence of potassium carbonate and potassium iodide in dry oxygen, and reflux this mixture for 18 hours. We have got the two. On the TLC, we got the two products, which both the products are separated by the column chromatography, and this compound yield is very low, so we could not proceed with this molecule, and we proceed with this compound, and this go the click chemistry. This is the alkyne and this is the azide. We go in the copper sulfate and sodium ascorbate in DMFM water, eco-friendly solvent. We got this triazole in one spot. We got this bis nucleus contain this triazole containing triazole compound. This R is a different groups present here, and this paper is published in 2013 in European Journal of Medicinal Chemistry. And this is another compound which I have already told you. These are phenylalanine diamine derivatives, in where we have used phenylalanine orthophenylalanine diamine, and we selective protection of this amine group by Bock in triethylamine, and in methanol at room temperature we got the Bock protected compound, which on propagation in triacetone we got the two products. The yield of these two products are very good enough to proceed further. So we have to provide this compound by column chromatography and use the click chemistry for cyclization of the development of the triazole nucleus. This is monotriazole containing compounds, and this is the bitriazole containing compounds. And this paper is published in Medicinal Chemistry Research in 2014. And this is a chalcone and triazole nucleus containing compounds. Here we have synthesized a chalcone and flavone derivative by using this 3,4-dihydroxybenzaldehyde and propylated it in presence of the sodium hydride and DMSO at room temperature. We got the three products. This is the isomers in where the propyl group goes here. In one it is gold here, and both the propyl propyl at both the positions. These three compounds are separated by the column chromatography, and then these two propylated aldehydes is treated with this 1-hydroxy 4 and 6 dimethoxy acetophenone, and we got the chalcone. Corresponding chalcone, we got this chalcone. These chalcones on treatment undergo the click chemistry with the different azides. We got the chalcone chalcone derivatives, triazole containing chalcone derivatives. Here we have cyclized this chalcone into the flavone. By treating this chalcone derivative by iodine and DMSO and reflux, we got this nucleus is closed and we got the flavone. This flavone, after synthesis of the flavone, we have to undergo the click chemistry with different aromatic azides. We got the triazole nucleus containing flavanone and triazole containing chalcone compounds, flavone compounds. This paper is published in European Journal 2016, and these compounds are showing the very good antibacterial activity against gram-positive and gram-negative bacterial strains. This is a derivatization of the molecule of ciprofloxacin, existing drug. Here we have to take in the ciprofloxacin hydrochloride from the market, and we have removed this hydrochloride group by the 5% treating the 5% aqueous sodium hydrogen carbonate, and we got the free ciprofloxacin, which on propagation in presence of sodium bicarbonate and DMF and state for 40 at 100 degrees centigrade, we got the dialkylated product, which undergo the click chemistry in presence of copper sulfate and copper sulfate and 5 pentahydrate sodium ascorbate. We got this nucleus. We got the bistriazole containing compounds. This bistriazole containing compound, this group is undergo the this group is for antimicrobial activity, responsible for the antimicrobial activity, and this is a linker. This is link, and this fragment is the ciprofloxacin fragment. Here we have the X. We have used this different 2,4-difluoro, 4-chloro, 3, so on, and we have synthesized about 21 compounds of this and tested all these compounds against the antibacterial agent, antibacterial strains, and developed the SAR. How these compounds, functional group, are changing the activity of these molecules. And this is the tetrazole. This is a tetrazole triazole containing compounds. This tetrazole is synthesized in my laboratory by using this lead chloride. Lead chloride in one pot, we have synthesized this from acetonitrile. We have synthesized this tetrazoles, and this tetrazole on propagation, we got the two products. This is two products, and this compound yield is good enough for proceed further. So we have used this alkyne, undergo click chemistry with different azides. We got tetrazole and triazole compounds. These are the different synthesis of the azides, how we synthesize the azide in each every scheme. So this is a simple anilines which undergo with the treatment with the sodium azide and sodium nitrate in presence of the SCl state for 3-4 hours, and the crude temperature we got the azides. These are the R's with substituent. We have used different substituent for synthesis of the this azide, because we required a number of the compounds of the same series to develop the some SAR relationship. These are the peptide synthesis in which we have used the benzothiazole nucleus, and we treated this benzothiazole nucleus with protected amino acids in presence of HOBT and DCC in dry DCM at 5 degrees centigrade, state 24 hours. We get the mono amino acid containing nucleus. This FMOG group is removed by the 20% piperidine in DMF. Either you use this method or TFA, phenol, or water, or thioanisole, or 1,2-diethylene diol. Any method you can use to remove this FMOG. Here we use a 20% piperidine in DMF, and we got this NH2. This FMOG has been removed, and then again we have this treated with the another amino acid, and then protected amino acid HOBT, and this is the same reaction condition we have applied, and we got the dipeptide of benzothiazole, and this FMOG is again removed from this compound, and then we got the dipeptide of benzothiazole, and these compounds are again tested for their antibacterial activity, and we got the good antibacterial result of these compounds. These are after synthesis and purification. How is characterize that our compound has been synthesized, our desired product is synthesized or not? We go for the H1 and C13 NMR spectroscopy. This is the NMR spectra of this compound, which is showing clearly that our compound has been synthesized. How? This is the CS2 group. These both have the CS2 groups in this. This is a bistriazole containing benzothiazole nucleus. This compound is the most active compound of my series which I have synthesized. And this is the CS2. This is both the CS2 groups, and this is coming here. And this triazole hydrogen, is coming here, and other hydrogens are, these hydrogen are coming here and all here. This indicate that because we have synthesized this triazole nucleus, so we have to find out that but these protons are coming or not. It indicates that our compound has been synthesized. And this is the tetrazole and triazole containing compound. Here this CH2 is coming here, and then this compound, this hydrogen and this hydrogen and this hydrogen, 3 hydrogen are coming here. This indicates that our compound has been synthesized. Our compound has been synthesized because these are the distribution peak of this compound. And this is the chalcone derivative of propagated chalcone derivative. In here, this methoxy group, both the methoxy, all 3 methoxy groups are coming here. And this this A group, this is the CH proton is coming here because this is a propagated proton, and this this C is coming, CCS2 is coming here, A is coming here, and D, the OH group of this coming here because it's going the upfield. This is a chalcone derivative of this. This indicates that our compound has been synthesized. And this is another flavone derivative of our series in which the dipropagated product is there. In there, the A, this is the propagated CH is both the CH is coming here. And B, this CS3 and this CS3 is coming here. The 6 proton is coming here, and this D. This is a peculiar peak of this D. It is coming here because it is not in the our chalcone derivative. And C is here. This is a CS2. The 4 protons are coming here. This indicates that our flavone derivative has been synthesized. And this is the another chalcone which is containing the triazole nucleus. This A CS3, this CS3 is coming here. Because this CS3 is different from this here CS3. These both CS3 B is coming here because it goes to the downfield because it is near to the chalcone ring. And this B CS2 is also here. And C is coming here, and D, this group is coming here. This indicates that our compound has been synthesized. And this is another derivative of the chalcone derivative. Here the similar thing is coming. See, this triazole nucleus C is coming here. This indicates that triazole nucleus is formed, and CS2 is coming here, and this A and A is coming, CS3 is coming here, and the B CS2 is coming here, and this OH is coming here. And this is this triazole containing compounds of the triazole, this chalcone derivative. In there, see this chalcone hydrogen where the triazole carbon is coming here. This is the 2 hydrogen is here, and the BCS2 is coming there. This is the 4 protons, and the A CS3, both the CS3 is coming here. This is the 6 protons, and D is coming here. This indicates that our compound is the peculiar peak of this compound is that that the triazole nucleus is formed. And this is the flavone derivative of the monotriazole. Here is the B, CS2 is coming, and the C is coming there, and A is coming there. This is the most peculiar thing of this compound. And this is the this is the bis triazole containing compounds. Here the A is coming here. This is 6 protons. Both CS2 is coming here, and this C11C is coming here. This is indicate that our triazole nucleus is formed. This is again the carbon C13 of, we have also characterized our compounds by the C13 NMR. This is C13 NMR of this chalcone derivative, monotriazole containing chalcone compound. This A carbon, methoxy carbon is coming here, and then C, this is a carbonyl carbon. This is a chalcone carbon, carbonyl carbon is coming here because it go downfield because near to the diene compound, diene ring, and this A is also here, and B is coming here. This is also the another derivative of the chalcone derivative. It is a A proton. This methoxy carbon is coming here, and then CS2 is coming there, and A is coming, C is coming, this C is coming there. This is a peculiar for that chalcone because it is differentiate between chalcone and flavone. And this is also the same bis triazole containing compounds. This also the methoxy group is coming here, and the C proton is coming here, and B, both the CS2 group is coming here. This is also the flavone derivative. This C is coming here, and B is coming here, and CCS3 is also coming there. And this is also the this triazole containing flavone derivative is set. This carbonyl carbon is coming here, and the CS2 is coming here, and A is similar to that of other protons, other carbon spectra that is coming here. And this is also we confirm a compound with the mass spectra. The mass of this molecule is 506.11. And after characterization with mass and NMR, C13 and H1, we have also go for the crystallography. We have crystallizes our compound in different solvent by slow evaporation process. And this is a compound, chalcone derivative, which is crystallizes. And the crystal structure is monoclinic. And the temperature 293 we have used. And then this is the our ring. This is the oxygen. This is the chalcone. This is the diene ring, and then this is a triazole nucleus. This is triazole. This is 3 nitrogens are there, and this is a fluoro group, and this is a methoxy group. It indicates that our compound had been synthesized. And then this is a structure of our most active compound of benzothiazole series. This compound is by this triazole containing compound in there. This is the triazole nucleus, both triazole nucleus. This is the fluoro group. This is a this is also in a monoclinic system, monoclinic form. And this is the another compound, chalcone, another compound which crystallizes is a mono triazole containing nucleus. This is the triazole nucleus which is contained, and this is also monoclinic in structure. Other groups CS3 group are coming here. This CS3, this CS3, and this CS3. This is the OH. And this is the another compound which is crystallizes. This is also triazole. This is a carbazole nucleus, which also shows this. This is a carbonyl carbon, and this is the Bock group, and this is the nitrogen. And this is the another compound, phenyldiamine derivatives. This also crystallizes in the monoclinic form. This is the nitrogen, and both are the oxygen. These are the oxygen, carbonyl carbon, and this is a Bock group, or this is the propyl groups. And after that characterization, we go for the antiplasmal reactivity of our chalcone and flavone derivatives. Our compound, one compound showing the against hepatocarcinoma liver cell line, and the hook cell 7 is a liver cell line, and this compound chalcone derivative shows a very good IC50 at 2.74 microgram per ml. And this is our antibacterial activity data. We have tested our compound against gram-positive and gram-negative strains. Gram-positive strain we use S. aureus, E. faecalis, E. coli, and gram-negative P. aeruginosa, S. body, and Q. pneumoniae. Our compounds are showing the very good antibacterial activity in comparison to similar to that of ciprofloxacin. This compound containing fluorochlorogroup shows activity at 6.25 microgram per ml against S. aureus. This is similar to that of the ciprofloxacin, and this compound is also showing the similar activity at 6.25 microgram per ml, and this compound also showing the similar activity which is similar to that of ciprofloxacin. And these compounds are also showing the good activity against E. coli. It is 10C and 10D, which is similar to that of ciprofloxacin. This compound is also showing the similar activity 10D, 10C, and 10D, which is also similar to that of the ciprofloxacin. The another compound is that 12C, which is containing difluoro, and which is also showing the antibacterial 6.25 at E. faecalis, 6.25 at E. coli, which is similar to that of the ciprofloxacin. This is also similar to the ciprofloxacin. And this is the other compound which is showing not good activity, because we need a number of the compound, at least 20 or 21 compound, to reach some meaningful structure activity relationship. That how our group changing in our group, the compounds are showing the activity. That's why we are preparing about the 20 or 21 compounds of a series to develop some meaningful structure activity relationship. These are the bar diagram which is showing that our compounds like this. This is our compound. This is 10A, 10C, 10D, 10D, and 12D, and the yellow one is ciprofloxacin. These are all compounds are showing the similar antibacterial activity and is compared to this our standard drug ciprofloxacin. And this is a bar diagram of our antibacterial, our most potent compound. We have tested our compound against 5 gram positive and gram negative bacterial strains. This is S. aureus, E. faecalis, S. typhi, E. coli, K. pneumoniae, and P. aeruginosa. This red one is showing our this is the ciprofloxacin, and blue one is showing our compound. It means that you can see this compound, the activity against P. aeruginosa is a similar MIC, but in other in this case pneumonia, E. coli, Staphy, and E. faecalis and oryzus, our compounds are showing the just half of the MIC in comparison to cephaloxin. Cephaloxin MIC is 6.25, and our compound is showing the MIC as 3.25 in each case.
There is a question in chat box.
Yes.
You have taken, if you want, Dr. Dimple Kumari asked that, ma'am, white triazole rings show antimicrobial activity, or other groups also responsible for it?
Yes, both the groups are responsible because we are using both the whole molecule for the activity. But 1,2-thiazole nucleus it is reported that 1,2-thiazole nucleus containing the 3 nitrogen atoms, it shows the better activity because all the molecules in the market are also having the 1,2,3 or 1,2,4-thiazole nucleus. If the nitrogens are more, it shows more activity. You see any drug in the market which contain the triazole nucleus, it shows the better activity in comparison to the drugs having no triazole nucleus. That's why we are using this triazole nucleus.
Okay, so Dr. Dimple Kumari, okay, it's okay. Now, ma'am, carry on.
Okay.
Sir, हमारे लिए तो totally new subject है, but anyway, I am learning a lot by this webinar.
Okay.
Something new I am learning by this today's session at least.
Hello, ma'am.
Yes.
Hello, ma'am. This is Dr. Dimple Kumari.
Yes. Tell me.
Ma'am, actually, I have made triazole by using azido esters and phenylacetylene.
Dr. Dimple, 1 minute. I have complete my.
Okay. No problem, ma'am. Okay. Okay, ma'am.
You write your question, I will answer you. Okay? After completion of my presentation. This is the antifungal activity of our compounds. We have also tested our compound against fungal strains, and then we found that some of the compounds, this red one is our fluconazole. This is a standard market drug, and blue one is our compound. So our compound, this is our compound. This is containing containing the fluorine group at the benzene ring, 2 position of the benzene ring, and tested against this common strains. We found that our compound they have a similar MIC in A. niger and A. fumigatus, which is 1.56 microgram per ml. But others, this is higher. It is higher in C. informis, C. cruci, Candida albicans C. clinical, and C. Candida propagalis. Now this is all about the synthesis of the new drugs or testing of the new drugs for antimicrobial activity. Now we have started some work in our laboratory by using this preparation of some nanocatalyst. So this nanocatalyst is very useful nowadays. We are using in some drug delivery system or some application in the organic synthesis or some transformation of the groups. So we thought that we have to sort of start some work of nanoparticles to use in drug delivery and some modification in the organic molecules or their functional groups. So we have selected this synthesis of this magnetic nanocatalyst and synthesized in our lab. So we have started this synthesis by the ferric oxide nanoparticles and use them. It is treated with ethanol in ammonium tetraethyl orthosilicate at 60 degrees centigrade, 6 hours at stirring. With the stirring, we got this the silica coated nanoparticles. This is SMP means silica coated nanoparticle. We got the silica coating is gone there. This is a pink one is a silica coating. And then this silica coating compound, the floating nanoparticles, again treated with aminopropyl triethylsilane in presence of the ethanol at room temperature 24 hours. We got the the amino group is on this silica, and with amino protected silica coated nanoparticle. And this nanoparticle is filtered out, and then it will be dried, and again it will be treated with the dicyanoguanidine, because I want that guanidine group on our nanoparticle. So we have treated this, and in ethanol and triethylamine at 60 degrees centigrade, we got this digonidine containing nanoparticle. It means that this is the nanoparticle, and this is the silica coating, and then this is the amino group, and then this is our guanidine group. On this guanidine group, I want to prepare the acidic catalyst. So we have to use the chlorosulfonic acid In dried dichloromethane, we got this nanoparticle at the rate of sulfonic acid group. It means in finally our nanoparticle having the outer layer of sulfonic acid. This is the nanoparticle, this is the particle, this is the silica coated, and this is the propyl chain, and this is the guanidine. And in bigonidine and bigonidine containing sulfonic acid, this is the 5 sulfonic group are attached with this bigonidine. This is guanidine group. This is the final nanocatalyst, which is the acidic in nature, magnetic and acidic in nature. After synthesis of this compound, this nanoparticle, we have dried, and then we have how it is prepared or synthesized or not. How we will check this one? So we have to go for that IR spectra, and then PXRD, and other TEM and SEM, all this. So first we have to do the IR spectra of this nanoparticle, how it is prepared or not. So see one by one that this is the magnetic nanoparticle, this is the ferric oxide ring, and here this compound ferric oxide ring, this is the 3360 band in IR shows the FeO presence of the FeO group. FeO group is present, it means that FeO group is present, this is the nanoparticle. Now we have coated the silica layer. Second layer is the silica layer. We have coated the silica layer. So band at 580 and 921 indicate the SiO-Si group. It means the silica coating is done on the nanoparticle. And then go the third one. Third one we have added the aminopropyl. We added the aminopropyl trithylsilane. It means aminopropyl chain is gone in the silica. This is the aminopropyl chain. How it is? Then it is indicated by that this band 3284. This is the for amino group. It is showing that our amino group is attached to the silica. This is ASMP. But the amino silica magnetic nanoparticles. This is for. And this is the magnetic nanoparticle. This is the nanoparticle. When we have done the sulfonic acid coating, so this band 994 and 1188, 1188 band nanometer band shows the presence of this sulfonic acid group on my nanoparticle. It means that we have started from ferric oxide, then ferric oxide ring is present, then we have added a silica, these are the silica ring, and then this amino group, and this is the nanoparticle, and this sulfonic acid, this is 994 and 18, indicate the presence of the sulfonic group. Now this is from the IR, it concludes that our nanoparticle has been synthesized. Now I have checked the purity and crystallographic nature of our compound, so we go for the PXRD, powder X-ray diffraction of our Fe3O4 magnetic nanoparticle and nanoparticle at the rate of sulfonic acid group containing compound. So we have gone to the PXRD, and we found that the 2 theta degree, they are a different drag the different peaks at 220, 400, 511, 440. This is a magnetic nanoparticle. This is Fe3O4. And then after this, after final nanoparticle coating of the sulfonic acid, we found that our nanoparticle is this is the extra peak. This is the amorphous silica, and silica is present. That it means shows that our nanoparticle is synthesized, fabricating all the functional groups. Then we have checked by this nanoparticle by the SEM and TEM. SEM and TEM, in SEM in or TEM indicate the shape, size, and the morphology of our nanoparticle. How is the surface? How is the shape? How is its size? So TEM is indicate the size of the nanoparticle. Nanoparticle should be below 100 nanometer always. So my nanoparticle is the size of this nanoparticle is 35 to 40 nanometer, and this shape is somewhat roughly spherical. You can see this is somewhat roughly in spherical. Roughly spherical shape they are roughly spherical. It means that in terms that our nanoparticle with the size is within the range, and our shape morphology is somewhat roughly spherical in nature. Now I have characterized fully with our nanoparticle. Now we have thought that how we use this nanoparticle in some applications. We have used some its application. So we have taken the primary amine in presence of the formic acid. We have used this nanoparticle and we synthesize this got the formulation of this primary amine. This is the aldehyde group at this position. In room temperature, at room temperature within less than half hour, we got this product in simple alcohol. At room temperature we stirred and we got this So we have used this primary amine, different primary amines. We have used almost 18 or 20 primary amines, or we got the good yield and recovered this nanoparticle in each every time. We have, after one reaction, we have to filter it and then again dry it and reuse again reactions. It is a reusable nanoparticle and its activity did not lose. It is same activity in the second cycle, third cycle, fourth cycle, and fifth cycle. So our nanoparticle is very good enough and it is reusable in the fifth cycle and having the same activity. So this paper we have published recently in the RSC Advances 2020. How then we have formula our amines, primary amines we have checked by the NMR. This is the NMR spectra of this is a methoxy containing formyl amine, and this CHB is coming here. It indicates that formyl group is CHO group is attached in our amine. And then third is the second one is this also, this is carbon spectra. This C carbon of this is coming here. And then B CH3 carbon is coming here. And we also confirm this spectra by the mass, and this is the mass spectra of this ring, and this is showing the mass spectra here, and the mass peak is coming at 151. It indicates that our compound has been synthesized. How our nanoparticle is working with the formulation of amine? How it is working? So then we have taken this nanoparticle. This nanoparticle containing the sulfonic acid group, outer layer, I have told you. And then this is a formic acid. This H is coming there. This is an electron, this H is coming here, and this become the O minus. This is O minus, and hydrogen is coming there. This become positively charged, and OH is attached here because the oxygen is giving the electron the hydrogen, and they become positive charge, and this H. Now the electrons from here, this carbon atom is going there. Because this hydrogen, because it is become the positively charged, it become the positively charged. This become the positive charge, and this oxygen is become saturated. Now this is positive charge. Then the amine is coming, and amine nitrogen has a lone pair of electrons. It is donating here. Then it donating here, it means it here, and then become the OH2, and nitrogen is coming here. Then this hydrogen is leaving, and this water molecule is going on, or this hydrogen is coming, this nanoparticle. So this water molecule is leaving, and this hydrogen is coming with there. So our formulation is formed, this NHS becomes CHO, and this other nanoparticle is again recovering, same, because this hydrogen is coming here. So nanoparticles remain as such. It goes in the reaction, and it started the reaction, and then formulation is completed, again back to the reaction. We have simply, this is filtered, or by centrifuge, we have to filter this nanoparticle. This is the methodology how the nanoparticle is working with our reaction. And this is some selected papers which I have published from my lab. This is the formulation. This paper is which I am showing nanoparticle. This is RSC Advanced, and this is interaction of chalcone and analogs which I have shown by the human serum element. This is in the luminescence, and this is the paper is communicated, and this paper is published in RSC Advanced 2019. This is also the human serum HSA binding, HSA interaction of our most active compounds. These are the all synthesized papers published from my lab, and these are all the funding. This is the university, and this is from UGC and UKD and Department of Science and Technology. From where we are getting the funding, we are writing the projects to carry out this research in our laboratory. And these are all the students we are working on the different problems and published papers. This student, now the nanoparticles, this student had worked on this nanoparticles and HSA binding and submitted the research recently and joined in the Baba University, Lucknow. And these are all our institute is the medical institute. So we have some MD students. We have to co-supervise these students in their MD dissertation. These are all the MD students which I have co-supervised. And this is our prestigious Kashi Hindu Vishwavidyalaya, where I am working. I am proud that I am a employee of this famous and Kashi Hindu Vishwavidyalaya. And this is the famous Ganga Aarti of Kashi. And thank you very much for sir your patience for to listen me.
कैसे अपने फर्स्ट फ्लोर से कि निकलने के लिए
Hello, participant.
Yes, yes, tell me.
If you have any queries to ask to ma'am.
Yes, yes, tell me.
Ma'am, good afternoon. I am Rama Shankar Singh.
Okay, sir, good afternoon.
Finance, and I am also graduated from BHU.
Okay, sir, good.
I have learned a lot new thing from your this session. Just I wanted to know which is a better thing, fluconazole, ketoconazole, and muconazole, because I am a diabetic and oftenly I am using.
Okay.
So what is your view on that? Which is the best medication for that purpose in case of fungal infection?
Doctor, very good question, but I am not a medical doctor. I am a
But
PhD.
anyway, but you have lot of good knowledge, so you might be interacting with the student and other persons also. So you have better exposure than me in this
Doctor,
matter.
generally the pupils are using the fluconazole.
It is commonly used, but I think all are the same family.
No, all are the same family, conazole family, but some compounds have some different actions in the body. They have the different actions because the
That's obvious.
pharmacokinetics and dynamics are the different for each and every person. The
But
drug is secreted
fluconazole
in
is a
your
very
body.
old. I think it's a old family. Now
Yes, 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 very old
label
family.
is coming. Level 1, 2, 3,
Yes,
like that.
yes, yes.
Fourth generation, 3 generation.
Yes, yes, yes,
Oh.
sir. Yes, sir. That's why we
that
need
way it
because it is a drug resistance. So we need a new drug.
Yes, yes.
That's why we are searching the new drugs for antimicrobial agents.
Still, fluconazole is popularly used, what I have observed.
Yes, sir. Fluconazole is properly used for an antifungal drug.
Yes,
An
yes.
antifungal drug.
Being a diabetic,
There is no
many,
substitute in the market, doctor.
yes, yes.
There is no
Certainly.
substitute of market. No, that time in the present time there is no substitution of fluconazole in the market because we are searching. We are not on the final stage of any drug.
No, obviously, the new research is always taking place in, especially in medicine.
Yes, sir.
That's, no doubt about it.
Yes, sir.
Thanks a lot, ma'am.
Thank you, sir. Thank
For
you,
your
sir.
good-
Dr. Dimple?
हेलो
हाँ, हेलो.
मैडम ये हम जानना चाहते हैं कि किसी मेडिसिनल कंपाउंड का अगर हमको काइनेटिक्स एंड मैकेनिस्टिक अध्ययन करना हो तो किस प्रकार करेंगे? हेलो
Sir, chemistry से related. हाँ, sir, this question is related to biochemistry. You can check by
Hello?
giving the drug by getting the. Hello. Hello. Sir,
Yes,
this is
ma'am.
related to. हाँ, ये ना आपका क्वेश्चन बायोकेमिस्ट्री से रिलेटेड है, तो आपको इसको बायोकेमिस्ट्री वाले से समझना होगा कि और ये आप उसको ड्रग देने के बाद जब ब्लड में इसकी ब्लड टेस्टिंग होगी, बायोकेमिकल एनालिसिस.
नहीं मैडम वो नहीं, हमारी बात को नहीं समझ रहे हैं. किसी मेडिसिनल कंपाउंड का अगर हम रिसर्च पर्पस के नॉलेज से पूछ रहे हैं, किसी
अच्छा.
मेडिसिनल कंपाउंड का काइनेटिक्स है मैकेनिस्टिक अध्ययन करना चाहते हैं तो मतलब कैसे होगा?
तो डॉक्टर साहब आप उसको
हाउ कैन आई
अरे डॉक्टर साहब आप किसी को उसको देंगे ना ड्रग को, सेल लाइन पे काम करेंगे या आप उसको एनिमल मॉडल पे देंगे, किसी पे देंगे ना ड्रग को. जैसे मैं जो सारी स्ट्रेन्स पर टेस्ट करती हूँ, बैक्टीरियल फंगल स्ट्रेन पे टेस्ट करती हूँ, उन टेस्ट करने के बाद उस बैक्टीरियल फंगल स्ट्रेन से उस ड्रग को एक्सट्रैक्ट करके निकाल कर उसका स्टडी किया जाएगा काइनेटिक्स कि वो ड्रग उसके तो हमारे स्ट्रेन को किल कर रही है, तो वो उसके किस एंजाइम पे जाकर उसका कर रही है और उस एंजाइम से उसकी सेल वॉल का बनना खत्म हो रहा है कि उसका डीएनए उसका जो है फूड बंद हो रहा है, क्या बंद हो रहा है. क्योंकि कोई ड्रग होती है डिफरेंट फैमिलीज की ड्रग, जो बैक्टीरिया और फंगस होता है उसकी डिफरेंट उस पर जाकर एक्शन करती हैं. कुछ ड्रग्स होती है उसकी सेल वॉल को करती है, कुछ फूडिंग को बंद करती हैं, कुछ डीएनए सिंथेसिस को बंद करती हैं. तो जब आप उसको ड्रग देंगे किसी को और ड्रग को देने के बाद किसी एनिमल मॉडल पे देंगे या आप स्ट्रेन में देते हैं, तो उस स्ट्रेन से उस ड्रग को एक्सट्रैक्ट करके निकाला जाएगा और उसके बाद उसका अध्ययन किया जाएगा कि इसकी ड्रग ने जाकर वहां पर क्या किस चीज पर बाइंड किया. You can also learn this technique by the theoretical way, by molecular docking. कि how our drug is working inside the human body. because you have to test the sub protein from protein data bank and dock your compound with molecular docking, and you find out theoretically that why our drug is doing like that. Some people are doing on molecular docking also. You understand my point or not?
सर कुछ कुछ तो समझ में आ रहा है मैडम, लेकिन ये बातें हमें समझ में ये आ रही है कि स्पेक्ट्रोस्कोपी मेटा IR-NMR या मास स्पेक्ट्रो
सिर्फ आपको ये पता चल जाएगा कंपाउंड बना है कि नहीं बना है।
अच्छा,
आपको उसका मैकेनिज्म सिखाएगा तो आपको उसको जो जिसको भी देंगे आप, जैसे हमारे एनिमल मॉडल में देते हैं तो एनिमल मॉडल से उसका वो ऑर्गन निकाल करके उसका वो एक्सट्रैक्ट निकाल करके फिर आप चेक करेंगे, या आप उसको देते हैं सेल लाइन में देते हैं तो वहां से आपको निकाल के देखना, आपको ये देखना पड़ेगा कि हमारी ड्रग ने जाकर क्या काम किया, यही पता करना है ना।
नेक्स्ट फर्दर मैडम ये कि किसी मेडिसिनल हमको कंपाउंड का नाम बताइए जिस पर हम कैनेस्टिक या मैकेनिस्टिक अध्ययन कर सकें।
सर कोई भी ड्रग ले लीजिए, आप फ्लुकनाजोल ले लीजिए, सर ने कहा था उसका HS से बाइंडिंग कर दीजिए ह्यूमन सीरम एल्ब्यूमिन से। आपको पता लगा, मैंने पेपर अपना अभी बताया था ना ह्यूमन सीरम एल्ब्यूमिन में कि हमारा फ्लुकनाजोल जाकर बॉडी में क्या करती है, तो आप फ्लुकनाजोल ले लीजिए मार्केट से और उसको जो है ह्यूमन सीरम एल्ब्यूमिन मार्केट में मिलता है उसको ले लीजिए और उससे बाइंडिंग करा के देखिए कि हमारी ड्रग कहां कहां पे बाइंड कर रही है, क्योंकि ड्रग का ट्रांसपोर्टेशन बॉडी में सीरम के द्वारा ही होता है ना।
तो मैडम इसको हाँ, तो इसके लिए मतलब किस इंस्ट्रूमेंट के किन इंस्ट्रूमेंट की आवश्यकता होगा? हमारा का इंस्ट्रूमेंट उतना हाई क्वालिफाइड है नहीं ना।
तो आप कहाँ पर हैं? आप कहाँ पर हैं?
हम उत्तराखंड में हैं, एक PG कॉलेज है उसी में। और मैं
आप ऐसा करिए ना डॉक्टर साहब, आप ऐसा करिए किसी अच्छे इंस्टीट्यूशन से आप कोलैबोरेशन कर लीजिए, है ना। उत्तराखंड में CSIR लैब होगी, कोई लैब्स होंगी, पालमपुर में लैब है CSIR की, वहां पे आप एक बायोकेमिस्ट्री डिपार्टमेंट में बायोकेमिस्ट से कोलैबोरेशन कर लीजिए और वहां पर आप अपनी ड्रग देंगे तो वो बता देते हैं।
आप यहाँ से नहीं कोलैबोरेशन हो सकता?
नहीं सर, हम सिंथेटिक केमिस्ट हैं, आई एम नॉट डूइंग दिस वर्क। मैं भी कोलैबोरेशन में ही करा रही हूँ काम।
तो कहाँ से और कोलैबोरेशन हो सकता है मैडम?
सर देखिए, आपको जाकर के बात करना पड़ेगा, कोलैबोरेशन इतने आराम से लोग नहीं करते हैं, आप जाकर थोड़ा सा उनको वो कन्विंस करेंगे तब वो कोलैबोरेशन आपका करते, क्योंकि कोलैबोरेशन करने पे क्या होता है कि आपको उनको वो भी देना पड़ेगा, केमिकल्स भी देना पड़ेंगे, उनका टाइम भी लगेगा, तो यू गो पर्सनली एंड डिस्कस विद दीज थिंग्स।
बायोटेक्नोलॉजी विभाग से संपर्क करना पड़ेगा।
हाँ सर,
बायोटेक्नोलॉजी से,
हाँ बायोटेक्नोलॉजी से भी कर लीजिए, बायोकेमिस्ट्री से भी कर लीजिए, बायोटेक्नोलॉजी से भी कर लीजिए। आप ये डिफरेंट कोलैबोरेशन करके आप काम कर सकते हैं, ऐसे तो नहीं पॉसिबल हो पाएगा। हम भी कोलैबोरेशन ही करते हैं जगह सब जगह।
तो लखनऊ वगैरह में मैडम नियरेस्ट बताइए। हेलो,
हाँ
मैडम नियरेस्ट लखनऊ या बरेली के आसपास बताइए।
लखनऊ में देख लीजिए, लखनऊ में CTRI लैब है, CSIR ITRC है, देख लीजिए लखनऊ में।
एक सर मैडम, आप तो लोग रिसर्च में काफी हैं, एक चंडीगढ़ से हो रहा था वो कह रहे थे कि केस मेहता साहब कि वहाँ बहुत सा अगल बगल मतलब हमारे क्या सुविधा है रिसर्च पर्पस का, क्या ऐसा बात सही है? चंडीगढ़ के आसपास काफी रिसर्चर है, इक्विपमेंट की सारी
सुविधाएं नाइट पर है डॉक्टर साहब, ऐसी बातें है, नाइट पर है, 2-3 है, आप देख लीजिए डॉक्टर साहब जाकर। मुझे उतना आइडिया नहीं है, मैंने कभी किया नहीं, आप देख लीजिए एक बार जाकर, है ना। यू कैन गो
सर पर्पस से मैडम क्या मदद कर सकती है हमें?
आप क्या मदद चाहते हैं ये बताइए।
मैं भी जैसे मेडिसिनल कंपाउंड पर कराते हैं तो कुछ मतलब हमें भी हेल्प कीजिए, आर्टिकल निकालने में कुछ रिसर्च का बच्चों को नॉलेज देंगे, बढ़ावा देंगे, और हम लोग
मतलब आप मुझे मेरा जो वो है ना, आपके पास नंबर वगैरह है तो आप मुझे कभी फोन करके बात कर लीजिए तो मैं आपको बता दूंगी डॉक्टर, ठीक है?
आप सर कांटेक्ट नंबर दे देंगे, उसको हम अनुसार
डॉक्टर लक्ष्मण सिंह जी के पास है, आप ले लीजिएगा उनसे, ठीक है?
ठीक है मैडम, थैंक यू।
तो आप हमसे बात कर लीजिएगा तो हम आपको बता देंगे डिटेल में सारा।
ठीक, थैंक यू।
तो ठीक है, हम आपकी हेल्प कर देंगे डॉक्टर साहब, ऐसा नहीं है।
मतलब वही चीज नॉलेज, ये सब रिसर्च पर्पज का पढ़ाई लिखाई तो वहीं जाता है मैडम, लेकिन रिसर्च बहुत पुअर है। हम लोग भी चाहते हैं कुछ इंटरेस्ट ले और आप लोग के सहयोग मिले।
हाँ, डिग्री कॉलेजेस में इतनी फैसिलिटीज नहीं, आपके सुविधा होती है जो कि आप रिसर्च बहुत अच्छे से कर सकते। आई नो, मैं जानती हूँ, हमारे यहाँ भी ये नॉर्थ में भी यही हाल है, हमारे यहाँ भी, वो यूपी में भी कॉलेजेस में नहीं पूरी प्रॉपर वो है उनके पास फैसिलिटीज, वो रिसर्च नहीं कर पाते हैं, वो तो मैं जानती
मैडम। हाँ, वो बात इसीलिए ना है, बच्चों की संख्या बढ़ रहा है, एडमिशन लेना चाहते हैं बच्चे, कुछ जानकारी चाहते हैं, और हम लोग के पास जानकारी एक्स्ट्रा है नहीं, तो जब लोग आप लोग हेल्प नहीं करेंगे तो हम लोग बच्चों को मतलब क्या दे सकेंगे।
सर आई एम रेडी टू हेल्प यू, ऐसा नहीं है, हम बिल्कुल तैयार हैं। आप इस जगह आप फोन नंबर लेके मुझसे बात कर लीजिए, मैं आपको बात कर लूंगी, बता दूंगी। ठीक है डॉक्टर साहब?
ठीक है, थैंक यू मैम।
डॉक्टर साहब, वो डॉक्टर डिम्पल कहाँ गई?
डॉक्टर डिम्पल, Dr. Dimple, are you listening?
Hello.
Hello. Dr. Dimple.
No.
I am Dr. Pranita.
Yes. Hello, ma'am.
I'm from Jharkhand, Bingwa Baba University.
Okay.
Very good session, ma'am.
Okay, thank you.
I have a query. How can I prove the formation of a triazole ring in click chemistry?
How can
I prove the formation of triazole ring in click chemistry?
Okay. Ma'am, you go for the NMR. You go for NMR mass. First, you go the NMR. NMR H1 and C13 NMR. I show you that the peaks. Hydrogen peak in triazole nucleus, triazole ring. This is a 5-membered ring, and this is a CH. One CH is there. This CH hydrogen is coming very downfield. It indicates that your triazole ring is formed, and then the C13 carbon. This carbon is coming very low field. This indicates that your ring is formed. Or you can check by the crystallography. You can go the crystal. You develop the crystal by slow evaporation and do the crystallography. I showed you the crystal also. That the whole molecule is coming in the same structure in the crystallography. You can go through that. All the 3 techniques you can use. Or if you have some problem, you can call me.
Okay, ma'am. Thank you, ma'am.
Okay.
मैम, आपने जैसे कहा कि कोलैबोरेशन एंड दैट, बट जनरली देयर इज ए प्रॉब्लम फॉर स्टूडेंट, हाई chemical compound having a costly impact. This is generally not supplied to the student. Generally, these type of complaint is coming from the research scholar. What I have observed.
Sir, iska to it is a one thing that you have to write the projects in different funding agencies and gain the money from that and then do the good research. We are also doing the same thing. We are writing the projects and we are collecting
the compound which is a costly, always a problem for the researchers in general.
Sir, I know. But that's why this funding is a big problem in research, sir.
Unless it is a specific project, always student face problem because in a BB also.
This is a common problem, sir. This is a common problem because research government is not funding so much to the for the research.
Especially this
facing the same problem, doctor. Sir, but we have to write the projects to different funding agencies with collaboration with other universities or the other colleges, and you get the big project, and then you do your research.
Student specific यदि grant मिले, that may be better in that case.
Yes, sir, students को grant नहीं मिलती, sir. Always get the
because
supervisor.
what generally university system में what I observe as a finance officer and lot of people, because I am more academic interest also after joining BBU, but generally student always come with this complaint about this non-supply of the high cost chemical compounds which affect their research.
क्या होता है ना कि सर फंडिंग के हिसाब से, according to funding we purchase the chemicals. According to the chemicals we have to purchase funding, we have to design the molecules. क्योंकि everything is depends upon the infrastructure and the money.
That's correct, but if you want a very good research and that, then there should not be such problem for supply of the chemicals.
Always the money, always the costly chemical or costly research is not the good. The research is come from your mind. How you
but
think.
It is many, I seen lot of students coming to me being.
Sir, my compounds are very low cost. Sir, my compounds are very low cost.
Maybe but in other cases also, because this is always a subject matter of discussion.
You are right, sir. I agree, sir. But you have, but one thing is that you write to different funding agencies to collect.
And especially those
grant
who are a
and
costly compound, lot of unethical thing is also going on. What generally student is complaining for that. कि
नहीं
सर
नहीं सर,
परचेज
ऐसा
तो
नहीं
इतना
है,
होता
costly
है
compound,
मगर
you can
सप्लाई नहीं
purchase
होता
from
है.
Sigma, from outside na, outside country, from abroad, Sigma, Eldridge, you can purchase from there. We are purchasing, but you
नहीं,
have to write the project.
almost some fund is earmarked, but this type of problem generally student face. Supply of chemical compound and all these things.
Student guide is the responsibility to arrange the chemicals and all the facilities to the student. If we have taken a student under his or her supervision, then this is the responsibility of guide to arrange everything. My students are working, I am arranging everything, sir.
But that is there, but guide has certain limit because the funding is done by the central level, na? Vice chancellor
Okay,
and
sir,
committee
you
and
have
this.
you have to limit the number of the students, not higher than 1 or 2 students. I have only 3 students, sir, because my quota is 8, but I have only 3 students because due to lack of space, lack of chemicals, lack of funding.
Obvious. No, it's obvious, no doubt about it. It's obvious. But my view is that there should be liberal approach for supply of that. That may be.
Sir, we have to write the Modi ji or Ramesh Pokhriyal ji. Release some grant, more grant to this.
Because a lot of students coming to me when I was a finance officer in BBU, they are always complaining for that. Sir, we are
Sir,
not getting.
You are not writing to the funding agencies or UGC to send the more money to our college or university or research.
That is also there, but there is different saga to narrate for supply of the particular project funding and all these things. Na?
Because my university always say that you write to project to different funding agencies, they collect the money, get the grant and do this.
That may be different funding agency, but UGC level, it's not a very, you can say, encouraging response for the student. What generally they are coming with that. Maybe different project authority may are giving with that with certain limit. So that may be useful in that way. But as a whole, general fund for university is not very encouraging for the research and all.
Yes, sir. Every university
except 2-3 organizations, what I know, like NIPER, you rightly, ITRC, and there may be better position for that. But otherwise,
NIPER and ITRC CSR labs. But
they are good.
Our universities are not CSI lab. Our universities are UGC funded because our
that is a constant. Funding is a constant because of the UGC. No doubt about it. Yes, ma'am. Very nice session, ma'am. I learned a lot.
Thank you so much. Thank you so much, sir.
Where is Dr. Pratima Kumari? Participant? Have you any queries?
Hello, sir. Lakshman, sir. It's me, Dimple. Sir, I have typed my question. Actually, I'm attending this session with my both kids. So I have typed my question. Now they are not allowing, sir. Sorry.
Okay. So you asked, ma'am? You asked a question to ma'am?
Sir, it is in chat book.
Okay. I will read it.
It is in.
Ma'am, there is a question from Dimple Kumar is that we have made.
Sir, send me question. Where is this?
This is in chat box, ma'am.
Chat box is, where is chat box?
You have to go in more.
Yes. Okay. Dimple Kumari question is where? Yeah. Ma'am, we have made triazole using azido esters with phenyl acetylene, ditetra and hexatriazole rings in one molecule. Will they show antimicrobial? Yes, Neha. Is Dimple? Yes, Dimple.
Dimple? Actually, she is with the 2 kids, so she is mute her mic.
She can. They will. These compounds will show the hexatriazoles and tetratriazoles. I think they show better antibacterial activity.
So, Dimple, I think you have satisfied the answer. And any other person, do you have any question and queries?
Yeah, hello, ma'am.
Hello.
Oh, yeah. Are you listening to me, ma'am?
Yes, yes, yes.
Okay, my name is Dr. MMH Numani, and I'm from Bihar. I would like to ask a question. Ma'am, you have worked with a microorganism in the laboratory, like you have talked about your nano catalyst.
Yes.
When it will be used with the human being who is suffering from particular that microorganism, what would be the side effect, or what would be the mechanism? At that condition, there will be two, host. That the one would be the human being as well as the microorganism.
Okay.
Then it would affect on the human being too.
Dr. Saab, I have not tested this one. I have just synthesized the nanoparticle. I have not
Okay,
tested
then it will have to pass through that phase before coming in the market. And
maybe, but I have not tested. I have not seen, not studied yet this one.
okay, then
Not
what
studied.
would,
Because just I have synthesized and characterized and do the application of this nanoparticle. Your point is very good, sir. Valid point. I will check again. The next time I will see.
yeah, because when any medicine comes in the market and after trial, there are so many trial by which they have to pass. So it is the big things. There is some sir has been talked about the ethical issues and like.
Phase 1, phase 2, phase 3, different clinical trials phases. Then the drug comes to trial. I have just synthesized in my lab and tested. I have not gone to other. I am only the first one trial, phase 1, not phase 2, phase 3, phase 4. This is a new drug development.
Okay, just you have developed it, and it has to pass through several more steps. Then it will be in the market.
It is the phase 1, phase 2, phase 3. Then your different volunteers they have checked to then come to the market. It's a drug come into the market take 10 to 15 years.
10 to 15 years. Okay,
10 to 15 years. That's why we are not coming. The COVID vaccine is not coming in the market because the time limit is very short.
Yes.
It will just pass through so many clinical trials, then come to the market.
Yes, clinical trials. I'm talking about the clinical trials. Yes.
And dynamics is different because your body dynamics is different. My body dynamics is different.
Okay.
Some drug is working in your body in different way. In my body, it is working in the different way.
Yes.
So if it gives you more side effects, then my body it gives another side effects. That's why it takes time. You think that Pfizer is giving the COVID vaccine to their patients in the UK? But they are facing the side effect problems.
Yes. We have not getting the we have not getting the exact side effects about that vaccines which has been made, because I have seen in the news and somewhere that the people who got this vaccine are facing some side effects, but I'm not getting that side effects in a detailed manner. Okay. Actually, I'm from biotechnology and I did bachelor in chemistry, so I have interest in both the field. Okay, nice to talk to you, and it was very appreciated presentation. I'm very much impressed by it. Okay, thank you very much.
So, any other participant? Do you have any question?
No, sir, no question. Very good session.
Okay, ma'am. So, that is my point of view. I discuss about. Ma'am, are you there?
Yes, I am here.
Okay. As such as, ma'am, you are working in the area of nanomaterials and nanotechnology. Someone want to ask any query?
I think Dimple has come.
Yeah, I seen that someone has come. Okay, ma'am. So can I discuss something about nanomaterials? If you allow to me.
Tell me.
Actually, the session is 5 o'clock. So then that.
We can discuss.
As we can discuss that with nanomaterials. The nanomaterials we have using this size in this same of nano size 10 to power minus 9. So we have lots of studies about the zinc oxide, titanium oxide, and magnesium oxide, and lots of particles are there, and nanotechnology are there. So I think that why this area is so focusing on nanomaterials, because each and everywhere, magazine, news, science, everywhere. In the case of, if we talk about the scientific reports and talk about the nature and talk about the accounts and chemical research, everywhere, if we show that they have seen the lots of paper, lots of articles in the field of nano medicine and nanomaterials. So how we correlate that, how nanomaterials may be useful in that medical field?
Doctor, your question is very nice. Nanomaterial means the particle whose size is below 100 nanometer. Nano. And then this is in this area in medical sciences. Sometimes we are giving the drug to the patient, like a cancer drug we are giving the patient. So cancer drug is giving with the patient, they are killing the normal cell as well as the cancer cells. Both the cells they are killing. That's why the patient become very weak or immunity is zero. We want a drug which go directly to the cancer cell, but not inhibiting the side normal cells. So nanomaterial is a particle in where we have loading our drug with nanomaterial, and this nanomaterial is directly going to the cancer cell, not affecting the normal cells. This is a major use of nanomaterial. This time is like that because cancer research is going the 2-3 types. Directly delivery of the cancer medicine, the cancer cell, not affecting the normal cells. Now up to the drugs, they are affecting the normal cells also. So patient has become very weak and immunity is zero. That's why this nano, or nowadays in Alzheimer's disease, they are using the nanomaterial to reach the drug to beta amyloids, where this Alzheimer's disease is responsible for the Alzheimer's diseases. In my lab, 2 or 3 students are working on Alzheimer's disease. They are giving the drug through nanocatalyst or through nanocatalyst to Alzheimer's disease, because it is brain barrier. It is crossing the brain barrier and reaching to the drug at that level, because its size is very tiny. So now the person are preferring this, or this nanomaterial is also using daily life also. Agrochemicals like that, everywhere this nanocatalysts are using. They are using the nanocatalyst to maintain our life quality, to maintain the quality of life for the cancer patient, all the patients. In Alzheimer's, this is the most frequently using this nanocatalyst. Research, a large number of research is going on this field. Then sending the drug to cross the brain barrier and reach the beta amyloid, or which is responsible for the Alzheimer's disease. In cancer cells, they are also using this technique to cancer cells, to develop the cancer cells, to kill the cancer cells.
Nanomaterial, ma'am, also nanomaterial have some toxicity. So they also affect our organs.
This is also toxic. I have prepared the nanocatalyst of lead chloride, and lead chloride is toxic. The question from editor is this: this nanocatalyst is called, this nanocatalyst of lead chloride is called this is toxic.
Yes.
It remains, if it remains with the compound, then what will you are giving to the patient? What will happen? Is a toxic side effect going on? So we have to check that how much quantity we are using the nanocatalyst in the reaction and how much we are getting to the after reaction. In this way, we are finding out that our nanocatalyst is not with our compounds. This is catalytic sometimes, but it go to the fine stage. Its toxicity will be lost. That's why it is size is 35 to 40 or below 100 nanometer. Always below 100 nanometer. That's why. Because it is fine particle. Its activity increase and toxicity become zero. You have some idea about bhasma? Some Ayurvedic?
No, ma'am.
There are in Ayurveda, there are metallic preparation. This is called bhasma. In my faculty, this work is going on. This is the Ayurvedic department. Ayurvedic work is going. They are preparing the metallic bhasma. Metallic bhasma means, they say, suppose if you have the anemic patient, you have a anemia. You want the iron tablet, you will take the iron tablet. But there is a iron bhasma. They have take the iron ore or iron, and then they prepare the bhasma. The particle size of bhasma is below 100. Very bhasma is titrating with some plant drugs, plant extracts, and they're titrating it, and they're preparing the coordinate or chelate compounds. In that form, the iron become, if iron is ferrous, it becomes changing the ferric. And then they are giving it, heating it, after titrating, they heating it, and they're preparing the bhasma. This is the shelf life infinite. Otherwise, you will take iron as such, the metal, it is toxic. But in the bhasma form, it is beneficial to our body, because it is size become nanomaterial, and its viability is, it is increasing the viability of the drug, and is going the smaller to smaller part of our body. The same thing is nanoparticle, because if we are preparing the nanoparticle and we are attaching the molecules on the functional group on that, and then prepare, the size of nanoparticle become below 100 nanometer, because it is fixed. The size will be 100, below 100. If above 100, then it is not the nanoparticle. It is always below. The same type is in the bhasma of the Ayurveda. Because our Vedyas are using this bhasma. If you heard about that, our Vedyas on some plant, you are eating the Chavanprash?
Yeah.
Chavanprash is, what is Chavanprash? It is a Ayurvedic drug. It is a combination of 28 drugs. It is prepared in quantity by the Ayurveda. This is Ayurvedic drug. You are using the Tifla Churna. This is also the Ayurvedic. They are preparing from the plant and giving to the patients. Nowadays, you are thinking that Ayurveda is more flourishing than the modern medicine. Why? Because this Ayurvedic drugs have no some side effects. Because this is prepared by the herbal or Ayurvedic plants. Baba Ramdev is preparing mostly the Ayurvedic drugs from the herbal. They have the herbal garden. They are checking the quality and preparing the drug. Nowadays in COVID days, you are telling you to take the Bithania Ashwagandha tablet, Giloy tablet, Tulsi Ark you take. For what purpose? This is the immunity booster. They boost our immunity. Giloy is the immunity booster. Nowadays, people is planting the Giloy plant in their garden due to immunity booster. And Ashwagandha is also the plant. It is called the Indian ginseng. It is memory enhancer. It is hemoglobin, increasing the hemoglobin percentage. It is good for health. It is good for the diabetic patients. Internal strength, increasing the internal strength. Vithania somnifera ashwagandha is a very good plant extract. This is using for that. Baba Ramdev and the China is also using the plant. Haldi, curcuma longa is very beneficial to our body. Nowadays, it is telling that in COVID days, you have to take the golden milk once in a day, golden milk. For what purpose? Haldi is also a very good immunity booster. In anti-inflammatory, it is subsiding the virus or bacterial infection to our body. That's why telling that you will take the golden milk. You take the sun bath. Why? Because vitamin D is coming in our body, which is strengthening our immunity. I am working in the Ayurvedic department, Ayurvedic faculty. So they are using all these things. I am also taking the ashwagandha and the Giloy tablet daily for immunity booster. Giloy has some chemical constituent which is enhance our immunity. Ashwagandha has so many things which are, if you take ashwagandha daily tablet, you experience that your memory is increasing. You learned short some time. I am feeling like that. Ashwagandha. It is tablets available in the market.
has been okay.
At any medical shop, or say, give me the ashwagandha tablet. It is come from Ramdev, Himalaya. Himalaya is better. And then you can take from other Jhandu. The other pharmaceutical companies which are preparing this, Himalaya is the best one.
Himalaya is the best.
Himalaya. And Giloy tablet. The Giloy is also is very good for immunity booster. Nowadays, people are taking, they are taking the direct stem of the Giloy plant and then crushing it and boiling in the water, just half water they take. This is the strong immunity booster. So this Ayurvedic form is like our nanoparticle.
In the case of
using lots of years, hundred of foreign before Vyadyas and Rishis are using this basma due to nanoparticle. Going in the body, it is not harmful our body. Same thing is with
there is such example like Sona Chani Chavanprakash.
Yes, yes.
Yes, there is advertisement Sona Chani Chavanprakash.
Costly, but they are using the somewhat percentage of Sona or somewhat of silver, gold and silver.
Yeah.
Because silver and gold have the property. They have the property to prevent you from the cold immunity booster. Immunity booster. They are preparing also the bhasma from the pearl, shank, because they contain the calcium. They are taking the calcium tablet. They are using they are taking the calcium from this one, pearl, and then shank. They are using this for this purpose. Sona Chandi Chavan is that it means some quantity of Sona and some quantity of gold and silver is there. But they are, I think they are not using nowadays because gold and silver are very costly. But they are telling that this is Sona and Chandi, like that they are telling. But exactly everything is not available nowadays because some plants and some eco-type changes are coming in the climate, and that's why the genotype, genes of the plants are also changing. The quality of plant is not like that which is written in our literature by Shushruth or Charak. It is not available like that because everything is on hybrid. And the eco-type changes is coming, that the genotype is changing. You know very well that eco-type changes is coming, then the climate, then the gene of the person is changing. See the person of Assam, Arunachal Pradesh and Kerala and here, all they have the changes in morphology, the shape and size and everything is changed because due to eco-type changes. Arunachal Pradesh have small eyes, Kerala people are dark. Why? Because of climatic or eco changes there. That's why it is coming.
In the case of nanomaterial, these particles have a very density high, and it is very hard to attaching the drug molecule or drug carrier because this particle is small in size, and these are the separate one. So how will we modify the surface of these nanoparticles, or how will we bind these nanomaterial particles to drug or any other molecules? And in 2016, Professor Stuart from the Stanford University, he got the Nobel Prize in the area of microsynthesis or nanosynthesis of the macromolecules, such like that. They have seen that the molecules, if we fit the nanomatter inside the because the cases have of a macromolecule like that, a drug molecule, they have a cases, and they have a length like 2 nanometer, 3 nanometer, and they put these nanoparticles inside it. So these carrier and how these drug bind with the nanomaterials.
Actually, I show you the slides now that we have coating the different functional groups on the nanomaterial. Yes, I have prepared the sulfonic acid nanomaterial. magnetic iron is there in center, but it coated the different layers, functional group layers. So if they want to bind with this drug, so we have to, suppose we want to some drug in the mud, the drug has the OH group. Then our nanomaterial has some different group on the edge, outer layer, then it will both bind. Otherwise, it will not be bind. So, the nanomaterial, they have to change. If they bind this drug to our nanomaterial, then you have to develop that outer layer functional group on the outer side of the nanomaterial, which bind with our drug. Suppose I have shown you the slide that I want the hydrogen atom to our molecule for formylation. So I have attached the sulfonic acid group on the outer layer of the nanomaterial, from where we can get the sulfur. Accordingly, you can modify the nanomaterial that can be fit in our used as a carrier for our drug delivery. One student of my department is working on the Alzheimer disease. He is using the nanomaterial, liposome nanomaterial, and for binding the drug and giving to the patient. It means that this drug he is using, it should bind with the nanomaterial, which is the outer layer of that nanomaterial. That particular group will be attached to that nanomaterial that can bind with the drug. This depends upon the nature, which type of drug you are binding with the nanomaterial. Otherwise, you cannot drug go to the brain because it bind with the nanomaterial, then go. So we have to attach that particular group on the nanomaterial outside according to our drug binding. कि किस तरफ से हमारी ड्रग को बाइंड करना है तो नैनोमटेरियल को, तो हमें वो ग्रुप अटैच करें तो इजीली बाइंड हो जाए.
Okay, ma'am. So this is the very good point of view. So thank you very much, ma'am, for your nice and informative talk on medicinal chemistry. As we know that without chemistry, the existence of medical is not possible. I think that because in the current scenario we are saying that there is a COVID. So most of the scientists and researchers are only focused to develop the vaccination. So these are areas are importance in chemistry. Without basic chemistry, we cannot do we cannot apply into the medicinal chemistry. So medicinal chemistry is a simultaneous field as it links maybe scientific discipline and allow collaboration with other scientists in research and developing new drugs. Medicinal chemistry apply their chemistry training to the process of synthesizing new pharmaceuticals and also new drugs. So thank you very much, ma'am, again, and I am thanking to on the behalf of organizing committee on myself. This lecture and this talk must be informative to research scholars and our faculty members, and they must learn most of the things in the area of medicinal chemistry. And we will also thank our organizing secretary, Professor Kumar who is today join us. So thank you so much, ma'am.
Thank you, sir.
And, dear participants, tomorrow there is a very good session from the research professor of Dr. Satendra Singh. He is a research professor in South Korea, and he is talking about the basic field of the XID diffraction method and their useful. And he is also using the very technical tool like that expert and other cell and other software to how to define the crystallic structure, what is the plane, what is SKL, and lots of area. He's also defined the TAM analysis, how we will do the TAM analysis, how we will define the diffraction pattern, and lots of things should learn tomorrow. And we will join at 3 o'clock. And I see that the most of the participants wanting that please provide the feedback link. The feedback link we should provide on the last of the day of this program, and also along with the cues. And the cues will I already told you about that. The quiz must be based upon the query and question